This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 521, recorded on November 23rd, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Happy Thanksgiving.
Thanks. Same to you. It's a beautiful blue sky, sunny day here.
It's blue sky here. It's pretty chilly, though. It's 0 Celsius.
Oh, we're at 4.
Not too far off. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's good to be back.
Actually, you're not in Madison.
I'm not in Madison. I'm currently in my family's house in upstate New York, where it is a very cold -3 Celsius.
How many miles north of New York is that? 60, 100, 120?
Oh, probably a couple hundred.
Is it north of Poughkeepsie? Yeah.
Yeah. Yes. It's near Utica and Rome, in Oneida County, north of Syracuse.
Wow, it's pretty far.
It is.
And you drove by yourself?
I did. I drove Tuesday night.
But you're young. You stay awake. You won't fall asleep.
Yeah, I'm a night person. That's not a big problem.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We got 73 degrees here. That's 23 Celsius and mostly sunny skies. It's a nice day. A little breezy, but it's a nice day. It's good.
Day after Thanksgiving, we are dedicated to teaching you virology.
I'm not dedicated to going shopping.
That's true.
Yes.
Hey, wait a minute. Brianne, I want to, where exactly in upstate New York are you? What town?
The town is called Camden.
Camden.
Mm-hmm. It's where I grew up and where my family still lives.
And there's also Camden, New Jersey, of course.
There is a Camden, New Jersey.
There's one, Alan always says, every state has a Springfield.
Most states have a Camden.
Are you near the Erie Canal?
We are north of the Erie Canal.
Okay.
You know, your connection is better than at Drew.
Oh, well, that's good to hear.
It's clearer. Are you on a Wi-Fi network?
I am.
Wow. That's impressive. I guess we know you're in the middle of nowhere because there's nobody sharing your bandwidth.
Yeah. Oh, very true. I'm definitely in the middle of nowhere.
This episode is brought to you by the graduate and postdoctoral programs at the Department of Microbiology, Icahn School of Medicine at Mount Sinai. Composed of over 20 virology labs, all centralized in one building in the heart of New York City, this department is a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective grad students to apply to the program by the December 1st deadline. Interested postdocs are also encouraged to contact faculty of interest. For more information about the department, please visit bit.ly/micro-MSSM. December 1st. That's next Saturday. So this will be the last ad because the next episode won't come out until December 2nd, and it'll be too late.
Mm-hmm. I need to remember to write my recommendation letters for people applying to that program.
Yep. I also want to ask you to subscribe to my YouTube channel, youtube.com/PROFVRR, or you can just go to YouTube and search for Vincent Racaniello. Because as you will hear momentarily, me telling you PROFVRR apparently doesn't work. And here we go. This is a follow-up from Sophia, who actually left this message on the website. And I'm reading it because it does have some information for all of you, and not all of you I know go to the website. Dear Vincent, this is a message to help you increase your subscribers on YouTube. I honestly don't see a reason why you can't reach 25,000. Neither do I believe your listeners are not subscribing because they just don't care. I just think the way you tell us is not as practical, and people forget. These are my suggestions. 1, don't tell people to find you on YouTube with your initials. Just tell them to Google your name on YouTube. That's how I found you. I could never remember what you said by the time I had to look for your channel, but you can remember my name. Okay. 2, remember that most people are listening while doing other things and are not in front of a computer. So by the time the show is over, they forget. Why don't you tell people to subscribe just before you say goodbye so people remember and don't forget? It works. That assumes that everyone listens to the very end. I just don't know. I just don't know if that's true. Did anyone hear the Easter egg last week?
Oh, yeah.
Yes.
That was really, I thought it was so good. I had to do it.
I'll have to go back.
Yeah, you see, Condit listened to the Easter egg.
No.
You were the last person to say something in the Easter egg, actually.
Well,
I also put an Easter egg onto the David Quammen TWiV because we had a great talk before we started.
Yeah, I heard that too.
I put it at the end.
That was a great TWiV.
You like
I
that?
really enjoyed that. Yeah.
Yeah, it was fun.
Yeah.
All right, number 3, you need to sell your channel. Don't just tell people to subscribe. Tell them what they're missing by not doing so. I just found out that your channel has great stuff like the parasitic diseases lectures and virology in Spanish. 4, why don't you do a competition and give out something like a TWiV mug for everyone who subscribes by a deadline? That could be expensive because I need like thousands of subscribers more. I can't afford that.
Well, maybe you give it to the 25,000th one, or you have some drawing for people at each increment.
At each, we could do each increment. Yeah. 5, advertise your channel on your Facebook page so it comes in our feed and on your website, say next to the Patreon link so people can see it and click on it before they forget. Those are all good suggestions, so I will do as many as I can. Please do talk about the weather on TWiV. If people don't like it, they can skip and fast forward. It's not a live show, so why are they complaining? Good luck. Sophia is from Greece.
Excellent.
Well, the last week said he drives and he doesn't want to fast forward because he'll hit an abutment. And that's what Rich said when he was quiet at one point. I said, why are you quiet? He said, I just want to make sure I don't hit an abutment.
I listened to episodes of TWiV and TWiV on my drive to Camden, and I did not hit any abutments.
You probably didn't fast forward, right?
I did not.
Yeah, I don't either. I listen through the ads. And the podcast I listen to, the ads are actually entertaining for the most part, right? Mm-hmm. And they go on for minutes and minutes and minutes, but I actually listen to learn how to do ads properly. Rich, can you take the next one, please?
Trudy writes, hi, Twivers. Quick question. Are any of the people who complain about the weather small talk actually TWiV patrons? With a little winky smiley face there. Just a thought. Trudy. P.S. It's current. By the way, it's currently 48 degrees Fahrenheit and cloudy here in Atlanta, Georgia.
I don't really know. I didn't look. I don't know that. I could see that patrons or not, they could still not like it. You know, you're entitled to express your opinion. It's fine, and we're entitled to kind of do what we want, I guess. I don't want everyone. I mean, I don't want everyone to leave because of the weather, right? So we'll see. The numbers do not go down usually. Kathy, can you take the next one, please?
Sure. Anna writes, hello, Team TWiV. I wanted to respond to Dickson's comment on a recent TWiV that MCAT scores are predictive of graduation in medical school. Even if MCAT scores did predict likelihood of not completing medical school, that would be a fairly meaningless prediction. The non-graduation rate for medical school is very low, 4% according to the AAMC. In part because once an individual has been admitted to medical school, the school itself is invested in seeing them graduate. A more meaningful metric would be whether MCAT scores can predict a person's board scores. For instance, the USMLE Step 1 and Step 2. So there's the two boards that medical students have to take. Since board scores more closely determine a person's likelihood of getting into the residency program they want to get into. I realize that using a standardized test to predict outcome on another standardized test is not great, but it's the best proxy we've got. Studies have found there is no correlation, and Anna gives one link, and then another study that has a negative correlation, another link, or at best a weak correlation, a third paper. between MCAT scores and board examinations. There is also some concern that the MCAT is a racially biased metric as well, although that is controversial. I'm not advocating that MCATs be removed completely from med school admissions. After all, a good MCAT score could provide redemptive value for a student who struggled with grades early in undergrad. But I think the exam is sometimes given more weight than is appropriate, and sometimes is used inappropriately as a screening value with no other context considered. Anecdotally, I personally have only known of 3 people who were asked to leave medical school. I do not know the circumstances for 2 of them, but the third was not permitted to progress after failing, multiple times, a class which required showing empathy to patients, which an MCAT would not have predicted. It is snowing beautifully here in Madison at a reasonably comfortable 29 degrees Fahrenheit. Happy Thanksgiving to you all, and to those outside the U.S., Thanksgiving is a holiday in which the States celebrates that a long time ago, some immigrating asylum seekers made it to America and were graciously helped by the folks already here. Best, Anna.
Unfortunately, the folks already here were then slaughtered by the asylum seekers.
Yeah.
Which is not great. And I always feel badly every Thanksgiving, right?
Slaughtered and or relocated.
Yeah. It's too bad Dickson wasn't here to hear this.
But you can forward this email to him, right?
Yeah, well, he's actually was on the show notes earlier, yeah, so he's seen it. He's having Skype problems, so he can't, and he decided not to come in, and his Skype doesn't work. The moral is that if you decide not to come in, you have to make sure your Skype works ahead of time.
Yes.
I had not heard that it was a predictor of medical school, but many people tell me it's a predictor of passing the boards, as Anna says here. And that's why they continue to depend on it. Unfortunately, yeah, many schools do screening right outright. Above a certain MCAT, you get considered, below, forget it.
We, in the few years towards the end of, I served on the medical admissions committee at University of Florida for many years. And the last few years, they did not, how did this work? We did not, when we were discussing students, and I think even when we were interviewing them, we did not know their scores. So the staff decided who to interview, and I'm sure that they, at least to some extent, took the scores into account. But we didn't know grades or MCATs. That's right. We didn't know any of that stuff. And occasionally there would be a leak in one of the recommendation letters. But the idea was that we should evaluate these people just on their biography. And you could see what they had taken, but you couldn't see what their grades were.
Right.
And then, in fact, even we'd get together and discuss everybody, right? And it was kind of like a study section where you have a bunch of people in a room, each of who have interviewed a few of these folks, a couple of people, but not all of the ones that are going to be discussed. And so the people who'd interview them would introduce their records and discuss them, and then there'd be more discussion, and then you take a vote. And what we would do is we would take a vote, and then they'd show us the scores, and we'd take another vote and see. And these all went as recommendations. There was another committee that was a subset of these people that made the final decision. But the idea was to de-emphasize the test scores and even the grades, and try and of course, I'm sure that, I don't know what the staff did. I'm sure that there was certain minimal standards in the screening. But it was a nice process. That was a good process.
That's a good process to hear about. I think that if more applicants knew about that, it might reduce a little bit of the stress that goes into the days or two before the MCAT, which we see a lot of.
Mm-hmm. Yeah.
Well, UF prided itself on being a very sort of human environment, and justifiably so. It was a really a family kind of place to be, and this contributes to that. It was good.
The University of Michigan program in biomedical sciences, which is the umbrella graduate program, has discontinued use of the GRE scores for admission. There was a huge study about it. There was the pro side and the con side, and they came to that conclusion.
That's good. I like that.
Very interesting.
A lot of schools seem to be dropping it right now.
I understand that some people can redeem for some poor undergraduate performance, right? So I happen to know someone who is studying like crazy, taking courses, spending thousands of dollars to get a good score because they had a poor undergraduate, right, experience. Mm-hmm. But it seems to me 4 years of undergrad versus, a couple of months of studying for the MCAT, I don't know how that can predict passing a board.
No.
It's crazy. I think, I don't like these standardized tests because they put all your eggs in a couple of hours or 8 hours, whatever it is. And I don't think that's right. I think 4 years of college is what matters.
There's another interesting phenomenon with the MCAT, and I'm sure the same holds true for the GRE, and that is that for any given individual, the scores are usually pretty constant. People who take the test numerous times, this is not always the case, but in most cases, people who take the test numerous times don't move much in their scores.
Yeah.
I'm not sure what that means, but it's there.
However, I do think it's interesting that they have a course on empathy. That's good.
That's becoming more and more common as well. That it was sort of how to relate to patients was an integral part of the curriculum at UF, and is becoming programmatic in many medical schools. I've talked before about my sister-in-law who died many years ago, who was a resident at Beth Israel, and there's a program that that fosters humanism in science that was basically a spin-off of the tragedy of her death because of the kind of doctor that she was. So
I had an experience with my mom years ago, 20 years ago. She had colon cancer, so she had surgery, and she's in her room. They brought her to her room, and I wasn't there yet. My brother wasn't there. She wakes up. Surgeon comes in. I'm sorry, you have terminal cancer, and left, and without anyone in the room with her, right? And I came in later, and she said, "He told me I'm dying." She was so upset. I mean, it would have been better to have us there, right? And to all that stuff, empathy. But this was an old guy, old school, no empathy.
Wow.
Move on to.
Have you seen the movie Wit?
No.
It's about it stars Emma Thompson. And it's about, I think this was a pick of the week years ago. It's about a woman who has stage 4 uterine or ovarian cancer, I think, who is talked into joining a clinical trial by one of her physicians, and basically it documents her experience. It's fiction, but it documents her experience as a parade of the least empathetic physicians and health practitioners you could imagine, and it's quite a movie, and that is shown to the freshman medical class at UF as part of this curriculum. They have courses on how to deliver the bad news.
I'm sure. Yeah. I mean, nobody knows how to do it without some training, right?
Yep, absolutely.
I wish they had a course on vaccines, how to convince your patients to get vaccinated, because apparently a lot of physicians do not, because they give exemptions.
Yep.
Mm-hmm.
All right. We do actually have 2 papers, but, we're chatting.
Wait a minute. There's one more. Another email.
Yeah. Can you take that, Brianne?
Sure. MJR writes, "Please ignore Andy from Rochester and other complainers and keep doing what you're doing. TWiV and the rest of the Twix family of podcasts offers the opportunity to eavesdrop on reasoned conversation between smart people discussing interesting things that matter, plus some less important but humanizing chats, e.g. panelists, local weather, and science-themed emojis. A pleasant contrast to contemporary social media content.
All right, great. Thanks, MJR.
Puts it well. Eavesdrop on reasoned conversation.
Yeah.
Mm-hmm.
I think that's part of the appeal of this. Fly on the wall.
Yeah, I mean, Andy from Rochester is not a scientist at all and just wants us to get to the... And there are other people who do not like to chat, for sure. I know that.
I thought he's some kind of engineer. Or astrophysicist or something, but yeah, okay.
Engineer, yeah. By the way, everyone's connection is great today. It must be that everyone's out shopping and the internet is deserted. So we have a lot of bandwidth. All right, so we have a snippet in the paper for you, and they're both very cool. The paper is perfect for a post-Thanksgiving discussion. It's about a giant virus. If you just had a giant meal, we can talk about a giant virus. And it is an in-depth study of Mollivirus sibiricum, a new 30,000-year-old giant virus infecting acanthamoeba. It was published a couple of years ago, but Dr. Condit this week showed some interest in it.
Yes,
and,
indeed.
We will discuss it. And the authors are from mainly France. We got Aix-Marseille Université Grenoble Alpes, CNRS. We got INSERM. We got the Centre National de Sequençage. I guess that's the National Center for Sequencing, right?
Mm-hmm.
That's an interesting one. Russian Academy of Sciences and a hospital in Marseille. And the first two authors have equally contributed, Mathieu Legendre and Audrey Lartigue. And, of course, what giant virus paper would be complete without Chantal Abergel and Jean-Michel Claverie? Oh, Dickson is waving at me. Let me see if I can get him on. Hold on. Hello?
Hello, Vincent.
Dickson!
You revived.
Hey.
You came back from the tomb.
Cool.
I did.
You sound good, too.
Finally got this effing thing to work.
All right, good. I'm glad that you persisted. By the way, Dickson is coming to us from Fort Lee, and that's why he's having issues, and he's not opposite me. Dickson, here the weather is 0 Celsius. Is it 0 there?
I presume so, but you've got a heat bubble, so it might even be below 0 here. Who knows?
That's right. All right, now get back to the paper. Mollivirus sibiricum. This is
right.
A virus pulled out of Siberian permafrost, and
a 30,000-year-old virus, right?
At least.
Okay. See, I read the show notes. I did all that.
And we've previously talked about another virus pulled out of permafrost in Siberia, the Pandora viruses. No, that's not Pandora. It's Pithovirus. Pithovirus sibiricum. And this is the second. And this paper is, in fact, from 2015. So, Rich, what got you interested in this? Can you tell us, or should I not ask?
No, I can tell you. Well, let's say I stumbled across it doing research for another topic.
Okay, that's good.
Okay, and but what got me interested in it is one figure in particular, where they do. They, in effect, fluorescently label the DNA. This paper is just a basic characterization of this virus. They pulled it out. They have electron micrographs of the virus. They sequence the genome. They do a proteome of the virions, find out what proteins are in it, and they do a basic characterization of the life cycle. And as part of that basic characterization of the life cycle, one of the things that they do is to effectively fluorescently pre-label the DNA in the virus particles, and then infect amoeba with those labeled virus particles and ask what is the fate of the viral DNA. And there are very clear pictures that show that labeled viral DNA goes to the nucleus.
Yeah, it's
Okay.
beautiful. It was very pretty. Yeah.
They're very, very nice. Okay. Now, why that's interesting is that this is a virus, and we can do the rest of the paper if you want, but I'm telling you why I was interested in it. This is a virus that's part of this group of viruses that are called nucleocytoplasmic large DNA viruses, NCLDV, and one of which, the baby one, pox viruses, I spent my career on. And for a large fraction of those viruses are characterized by the fact that they are DNA viruses that nevertheless replicate in the cell cytoplasm.
Mm-hmm.
Okay, that's a big deal. There are variations on that theme, but the variations, in my mind, sometimes are not very well characterized, and we can go into a little more detail. But this really struck me because it's absolutely clear from what I can see here that this viral DNA goes to the nucleus. Mm-hmm. And yet at the same time, the virus winds up making factories ultimately and maturing in the cytoplasm. And it encodes an RNA polymerase as if it could do some of its own transcription. But it does not package the RNA polymerase in the virions. The implication of that is that it could actually do the early phases of the infection using the host RNA polymerase in the nucleus. Mm-hmm. And then the late phases of infection, I suppose it could do in the nucleus, but transcription. But it could also potentially do it in the cytoplasm. If that's the case, then it very closely resembles the iridoviruses, another family in this large set of viruses. That there, the claim is that they do the early part of their life cycle in the nucleus, and the late part of their life cycle, both transcription and replication, in the cytoplasm. But I've never been, those are really old data, and I've never been 100% convinced. But when I saw this, I thought, wow, okay, this really suggests that for this virus, something like that could be going on. That's what caught my attention. Mm. And then I got just totally weirded out and started to ask this question about lifestyle of all of the viruses in this giant virus family. And I can go over that as we go through this.
And Rich, it's the figure 3 that you find really convincing, right?
Yep. With the green DNA.
Yeah.
Yeah,
first in the cytoplasm.
Yeah, it starts in the cyto, right? And then pretty quickly, 30 to 90 minutes, right?
Yeah.
It's in the nucleus.
It's clearly in the nucleus. Now, we did a paper on TWiV 440
right.
on pneumonia virus.
I hardly knew me a virus.
And there, the characterization was, now this is another example where the virus encodes its own RNA polymerase, but if you look at the proteome, the protein complement of the virions, there's no RNA polymerase in the virion. And so this would be a candidate, once again, for an iridovirus-like lifestyle where it needs an RNA polymerase to initiate the infection and would start that in the nucleus, but it also encodes its own RNA polymerase. So then maybe it moves to the cytoplasm as iridoviruses are supposed to do and finishes off transcription and replication. But in this particular case, the characterization of the pneumonia virus was that it goes in and the nucleus gets really totally wonky. Okay.
Right.
It gets all sorts of invaginations and stuff. And from what they could, they had evidence that proteins leak out of the nucleus big time. And they seem to think that the viral DNA stayed in the cytoplasm. And so their conclusion was that the transcription enzymes from the early phase are leached out of the nucleus into the cytoplasm and used for the virus replication. Once the virus makes its own polymerase, it can continue off, but that it never actually enters the nucleus. And I was thinking, man, that's even the same authors in years following the Mali virus paper. And I was thinking, man, why didn't they do this fluorescent DNA labeling thing? Okay. Because that would have been more convincing.
Yep, they didn't.
So
Does anybody know what the ploidy is for the host cell, acanthamoeba?
Mm-mm. Do you?
don't know.
No, but most protozoans have more than one set of chromosomes. So it's possible that there's an advantage to that with regards to the replication of this very large item that gets into the nucleus without disturbing the host.
I'm not sure it's the natural host.
Yeah.
I mean, they use
host.
acanthamoeba. What's the word they use to?
They didn't. It wasn't bait. But something like that.
Yeah. It was a good word, and I've lost it
Yeah.
here. Maybe we'll come across it. And they say this is the fourth large virus that they've gotten from. They basically throw the melted permafrost onto acanthamoeba. But I don't know in nature if that's the original host, right? So the cool thing here, let's start a little bit at the beginning. They initially saw this virus using light microscopy,
Yeah.
right, of the amoeba cultures that they've been inoculated with. The thought,
That's right.
this is, by the way, late Pleistocene Siberian permafrost. People have taken cores, and they date them, and they can share them. And I think in this case, they got a hold of some of the core and thawed it out and threw it on acanthamoeba. So they're 500 to 600 nanometers in diameter. So they're not the biggest of these giant viruses, but I like their description that the particles are surrounded by a hairy tegument.
Mm-hmm.
Mm-hmm.
2 to 4 25 nanometer spaced rings corresponding to fibers of different lengths, and there's a portal in the particle, which is presumably where the viral DNA gets out. There's also an internal lipid membrane, which is pretty cool. And they go through the replication cycle, as Rich alluded to. They use an MOI of 50. And I'm going to make fun of their English again. They use a very high multiplicity to warrant the synchronization of the infection. and Jean Michel, we made fun of him last time because he wrote encoding everything but the ribosome or something like that. And he wrote back and he said, I appreciate your nice criticism to warrant the synchronization. I don't think you need a MOI of 50, though. I mean, 10 should do it, right?
Mm-hmm.
It doesn't.
It
shouldn't matter. It doesn't matter. So the particles are taken up by phagocytosis. And as Rich said, when you label the DNA, it seems to go right to the nucleus. And the virus replicates the cell. It doesn't seem to be destroyed. The nucleus is not destroyed either, right?
Nope.
As it is with some of these other viruses.
Nor is there activity. They can still move about in the culture's milieu.
Actually, in this case, in the long run, is the nucleus not destroyed? It's certainly not destroyed.
It says it's the periphery of the persisting, albeit deformed nucleus.
Okay.
So it gets deformed, but it's not destroyed. It still exists there. 651,000 base pair double-stranded DNA. They did a transcriptional analysis to see all the protein coding genes. I love how they say some of these things here. Well, this is later. I'm sorry. I'm jumping ahead of myself. 523 protein coding genes encoding 3 tRNAs. And
that's incredible.
Well, that's nothing. Some of these others have almost everything, but the ribosome.
Sounds like a genome poor E. coli.
The proportion of proteins without a recognizable homologue was 64%. And among the ones with the homologue in the databases, 17% were similar to a virus protein, 9% to an amoeba Castellani protein, 4% to proteins of other eukaryotes, and 3% to prokaryotic proteins. This is a real turducken. No, not turducken. What's the stuff at the bottom of the lake?
That's microbial ooze.
No. You have a word.
You're not thinking of Schmutzdecke.
Schmutzdecke.
That's on top of the. That's on.
This is genomic.
Top of the on top of the what? The cesspool, right?
I thought it was at the bottom.
No,
it's a layer in a treatment plant. It's a layer in a water treatment plant.
Well, this is genomic Schmutzdecke.
Yeah.
Genomic Schmutzdecke.
7 enzymes related to DNA metabolism, 4 transcriptional components, some kinases, some ATPases, some capsid proteins. The sole mollivirus protein, remotely similar to a major capsid protein, was identical to a amoeba castellani protein. And it's the only large DNA virus without its own ribonucleotide reductase, which, of course, is absolutely needed for the synthesis of deoxyribonucleotides. Yes,
so they don't tell us what those other eukaryotes are that have similarity to some of the genes.
Mm-hmm.
I wonder if knowing what those are could give us some idea about the natural host.
Perhaps.
Right.
Perhaps. Or, in that Schmutzdecke, there's a lot of DNA floating around.
Right.
Just take it up. But, yeah, it would be interesting to know that. You could do it, Brianne. You could get the data and...
It's true, I could.
Put one of your students on it. It would be a good student project, right?
Absolutely.
Well, of course, you want them to work on your stuff. But.
Well, I could. And they probably wouldn't work on it at the end of the semester. They have a fair amount going on right now.
So in order to replicate, it's got to utilize the ribonucleotide reductase of the host cell. It's interesting. Then they look at the proteins in the particles. It's pretty cool. This has 230 different proteins in the virus particle, which they did by mass spec. 59% are from the virus. 41% are from acanthamoeba.
Host.
The host, yeah. So these are in the capsid. 50% of the virus proteins that are in the capsid are orphans. ORFANS, the open reading frame, doesn't look like anything. So they made a clever word out of it, right? Orphan. And 25% could be associated to proteins with function. The 7 components of the virus-encoded transcription, this relates to what Rich said, 7 components of the virus transcription apparatus are not present in the virus particle. And that's why presumably the DNA has to go in the nucleus, right?
Right.
Rapid migration of the genome into the nucleus. Here's another funny thing. There's a protein with the very nice name of ML347. It's a homolog to the major capsid protein found in all large icosahedral DNA viruses, but of course these particles don't have icosahedral symmetry. It's very curious, isn't it?
Very curious. Even for a non-virologist, it's curious.
So there are 3 categories of functions in the amoeba-derived proteome of the virus particle. And the first is 11 ribosomal proteins are in the virus particle, both small and large.
Those are from the host.
From the host. Then there is also mRNA binding and processing enzymes. And then there are proteins with similarity to actin or actin binding or cross-linking proteins. And there's a typo there. It's acting.
Yes,
twice.
yes.
He's got acting twice.
Right. The ribosomal proteins, they list I guess first they list the ones that have the highest abundance or something, but they say there's a total of 23 different ribosomal proteins altogether.
Yeah.
But no ribosomal RNAs.
No. So I wonder what they're doing there, if it's an accident or they're actually contributing.
Well, what they say is they think that the proteins are not from intact ribosomes, but maybe just come from the nucleolus, which is near where the virions are assembling in the nucleus.
Yeah, right.
Yeah, a lot of ribosomal proteins on their own are basically worthless, I think. Outside of, they aren't going to do anything on their own, or not necessarily even be, if they were delivered to a cell, incorporated into ribosomes. Okay, so it's not clear you'd have to go through protein by protein and figure out what their potential was for contributing to an infection. But I would guess that in lots of cases, that potential is close to nil. So,
yeah.
Do you think this virus is still alive? Not alive, but active someplace?
That's a point they make at the end that maybe it's extinct. But with the thawing of the permafrost, with global warming, it might be something that we'll see again somewhere.
It was just hiding.
But Acanthamoeba is ubiquitous, Dickson?
Yeah, pretty much so.
I don't know. I think people probably haven't looked all that much,
Well, you could look now, though, right? You have certain sequences here that are only going to identify this one. So
you could interrogate the genome.
Interrogate the Earth.
So here's the word. It is bait in the discussion.
Using
Acanthamoeba castellani as bait, we isolated a new type of giant.
You think they fish, Dickson? Maybe.
FPFHISH.
No, that's good. Very good. Do you know that word?
Things are very fishy.
So they do a couple of other notable points. One is that they do an analysis of the transcription throughout the. Actually, they do a proteome analysis throughout the replication cycle, which means you do mass spec at different times after infection. And they say, indeed, what happens within a cell at a given time is a consequence of protein abundance, not the levels of their cognate transcripts, which I think is funny because everyone does RNA transcriptional analysis because they can afford it, whereas not everyone has a mass spec that they can probably dominate for weeks at a time, as was probably the case here. But they basically give data that suggests that there are two phases of gene expression. You have a phase where clearly the virus is going into the nucleus and utilizing the nuclear transcriptional apparatus. And then there's a subsequent phase where it's probably happening in the cytoplasm by the viral machinery. Would you agree with that, Rich Condit?
Well, I looked closely at this because I was interested in this idea of whether the second bit is actually happening in the cytoplasm. And they're actually quite circumspect about that because they don't have any direct evidence. And what they see here is that in the proteomic analysis is that the DNA polymerase comes up at least an hour before the RNA polymerase subunits. Suggests to me that the RNA polymerase subunits that you're seeing synthesized could be what they call post-replicative. Okay, there's a class of transcript that the DNA replication makes the transcription in the nucleus makes early gene products that include DNA polymerase, and then once the DNA starts replicating, that opens up a second wave of transcription that would, in this case, account for the RNA polymerase subunits. But that doesn't make total sense to me in terms of what I'm thinking, because if you're going to make RNA polymerase and use it for the late phase, you ought to probably make it at the same time as you're going to make the DNA polymerase, thinking like a virus. It would be interesting to do this experiment in the presence of a DNA replication inhibitor. That's the classic experiment.
Right.
And find out whether the DNA polymerase is made and the RNA polymerase subunits are not. So I don't know if this is strictly a timing issue or if it represents different phases. As I said, it's clear that a lot of the assembly and stuff goes on in the cytoplasm, but they don't make any really strong claims about whether any part of the DNA replication or the transcription is happening in the cytoplasm. In the case of the iridoviruses, there's the model is that the virus goes in, sends its DNA to the nucleus, and there is early transcription and an early phase of DNA replication in the nucleus, and then it moves, that whole thing, the genome, moves to the cytoplasm, where there's continued DNA replication and continued transcription. So you have both replication and transcription going on in both compartments in sequence. It's not clear what's happening here. And in fact, in the case of the iridovirus, when we did the pneumovirus paper, and I was trying to figure out whether it was really the same as iridoviruses or different, whether it was really taking enzymes out of the nucleus. I looked at the data in both those papers and I thought, all of this stuff ought to be done again. and really pin this down. Yeah. That's why I was so interested in this, because if I were doing it, I'd use this fluorescent labeling technique in all of the other cases and get a clear picture of where the DNA goes. But in this case, they don't really have a clear picture, and I'm not faulting them. It's just not part of the paper, of where the late transcription happens.
The last experiment they do is to actually sequence their Siberian thawed permafrost sample. Deep sequence it to see how much of these viruses are in there. And they end up calculating 1.1 pithovirus and 2.7 mollivirus virions per A. castellani cell in the original sample, because they can find A. castellani genomes in this permafrost. So I guess that's good that it's there, because it grows in it, and it suggests that that might be a natural host. It could be others as well, but maybe that is as well. So that's pretty neat. So let's put this in a little bit of context. We have a whole bunch of different kinds of giant viruses. So viruses were the first ones. 1.26 million base pair genomes, 1,120 proteins. Then we have that's a family, Mimiviridae. Then we have Marseilleviridae. These are icosahedral particles as well. They have 346 to 380 kilobase genomes. Then we have the Pandora viruses, which they call the spectacular Pandora viruses, because they're 1.1 to 1.2 microns in length. And their genomes can be as big as 2.7 million base pairs. Those were found in water samples in Australia and off the coast of Chile.
But now those Pandora viruses, those are Phyco. So yes, those are the family is Phycoviridae.
Phycodnaviridae, right?
With a PH.
Right.
Yeah.
And then we have the pithoviruses, which what's the family of the pitho? The pitho and the mollivirus.
The pithoviruses are pithoviruses. The mollivirus is a phycodnavirus.
Phycodnavirus, okay. And if you're wondering on how these are related, so the molliviruses from this paper, if you do Dickson, don't breathe so heavily. If the hello, Dickson. Dickson, are you there?
He just died. He just held his breath and died.
Sorry, I'm sorry. Molliviruses phylogenetically cluster as distant relatives of Pandora viruses. But they say it's not clear if it's ancestral, if Pandoras are, if the mollies are ancestral to the Pandoras, or it's because so many Pandora genes have been inserted into the mollivirus genome. So these viruses are very prone to horizontal gene transfers, as you can tell by the listing of where the genes came from that we did earlier. And the other thing I think is interesting is that this issue of the nucleus, the nucleocytoplasmic viruses are the mollivirus, Pandora virus, and the entirely cytoplasmic are the pithoviruses.
Well, actually, I got a little summary here, because this is what interested me, was whether there's any sort of phylogeny to this lifestyle.
Right.
Okay.
Right.
So I tried to figure this out, and what I come up with is that the Mimi, Pitho, Aspha, and Pox are all strictly cytoplasmic.
Right.
The phycodnaviruses are a bit of a mixed bag, because that includes the chlorella viruses, which are strictly nuclear. They don't encode an RNA polymerase. They don't carry one with them. They're strictly nuclear. And then there's this mollivirus that at least spends the first part of its life cycle in the nucleus, but encodes an RNA polymerase, and so has the potential, at least, and it goes through its morphogenesis in the cytoplasm. So that has the potential, at least, for having a sort of a split life cycle. And the iridoviruses, according to ancient literature, have a split life cycle. That's a different family. And the Marseille viruses, that includes the pneumovirus. And that's the one that where, according to the paper that we did, the DNA doesn't, the virus encodes an RNA polymerase, but doesn't carry it in the virion. But the DNA does not enter the nucleus, but rather cuddles up to the nucleus and uses enzymes leaching out of the nucleus to do its early stages of transcription and DNA replication. But I don't see, so there's several lifestyles, and I don't see from what I can see in this any particular phylogenetic logic. Okay? They seem to be sort of crazy split all over the place. And that's why ultimately I wrote to Eugene about this, Eugene Koonin, because I was trying to sort it out in my own head. And I said, is there any logic to this? And he doesn't really see anything. The drum that he's beating is that he thinks that these viruses started out smaller, I would say, but probably he didn't say this, but I'm thinking cytoplasmic, like the pox viruses, and through evolution, they got bigger by acquisition of all sorts of stuff. But in the process, some of them lost things that gave them the complete autonomy in the cytoplasm and acquired a nuclear phase. That's his spin on the thing, but I think there's a lot of work to be done.
So do you think that these viruses co-infect a lot in nature, and that's how they exchange all these abilities between them?
I would guess so. The giant viruses in particular, a lot of their giantness, and I had a phrase highlighted in one of these papers. I'll see if I can dig it out. Relates to infecting these amoeba, which are basically garbage trucks of one sort or another, and there's a lot of potential for genetic exchange as a result.
Schmutzdecke.
Well, I think primordial ooze is a better name.
I like Schmutzdecke.
So there's a ton of different species of amoeba out there that they could try, and a lot of them are in culture. Amoeba proteus is the one I always think of as the first one I ever learned about when I was in biology in high school. Even in high school we learned about that one. I wondered what the host specificity range for these for the viruses really are. You could line up a whole bunch of these amoeba and just try to infect them all.
I bet they're doing it.
Yeah,
I would hope so.
My question is, I've been sitting here Googling for a little while. Has anyone taken these same types of permafrost samples and tried to infect something other than amoeba? Have people looked for phages or other types of viruses in these samples?
I would assume so. They have done the metagenome of these, right in this paper, but they only report these giant viruses, but I assume they found other things, right?
That's what I assume too. I haven't found anything in searching.
Jean-Michel, what else is in this thawed stuff? Right.
So here's the phrase I was looking. Eugene sent me a paper that apparently it was in proof when he sent it to me yesterday morning, and yesterday afternoon he said it had been accepted or was published. So this is Koonin and Uten, and it's in Faculty of 1000 Research. There's a couple of interesting sentences at the end of this, and this is about evolution of these viruses. Why do giant viruses get so big? Part of the answer is likely to be simply because they can. Apparently, the acquisition of Apparently, the acquisition of an autonomous, robust replication system by ancestral NCLDVs unlocked the door for genome expansion. Furthermore, protist hosts seem to represent a melting pot of gene exchange and acquisition, where the evolving giant viruses can capture genes from the host, the endosymbiotic bacteria it harbors, and other viruses.
Yeah.
So there you go.
Maybe.
And I like that idea that if you have a robust autonomous replication system and lots of room in your viral structure for messing with your DNA, it opens the door for expansion of the genome.
Yeah, right. All right. So, Brianne, they say in the last sentence they did not see, known pox viruses or herpes viruses in the metagenome.
Right.
But they don't mention anything else. So.
Right. Well, I just think it would be really interesting to try to put this on a few different types of cells. They say no pox virus or herpes virus families. Is there anything that could infect a human cell or bacterial cell? I mean, the phages seem most likely.
We cannot rule out that distant viruses of ancient Siberian human or animal populations could reemerge as Arctic permafrost layers melt and or are disrupted by industrial activities. Are they saying that there's warming going on?
Sounds that way to me. Vincent, is that a large genome for an organism of a single cell? 46 million base pairs?
Castellani?
Yeah. Do you consider that normal or big?
I don't think it's too big.
Okay.
No, I don't think so at all, but I will... I think it's, what is it, genome.gov? No, that's not what the one. Giantvirus.org. That's not going to give me the cell. I was looking for, there's a list of cellular genomes.
I think the largest is orchid, if I recall.
The largest genome?
Yeah, because it's triploid.
Ah, well,
plants do that.
Strawberry is octoploid, I think.
Octoploid.
Well, anyway, I can't find it, but I don't think 46 million is particularly big. No.
All right.
Okay. Anything else on that? That was cool. Thank you, Rich. Thank you, Rich.
Thank you.
Giant viruses are always fun. And now for something totally different, we go from very big DNA to somewhat smaller RNA virus. And this is a paper from Current Biology. Collective infection of cells by viral aggregates promotes early viral proliferation and reveals a cellular level Allee effect. And this is by Ivan Andrew Moreno and Rafael San Juan from the University of Valencia. 2 authors, man. Just like the old days. This is like harking back to the olden days when, and Kathy and me and Rich and probably Dickson, we have had 2 author papers, but very rare nowadays. I thought this paper was cool. First, for one, I picked it because one, I knew Kathy would like it because she used to work on VSV. And secondly, it illustrates how you can take a relatively simple experimental design and get a ton of cool data.
Yeah.
Right?
It's also a really nice, simple question.
It is. The question is very good. So it revolves around this idea that, we kind of think that single viruses infect cells, but there's increasing evidence now that multiple viruses can get into cells, and some have been published. For example, lipid vesicles can have multiple enteroviruses in them. Marseille viruses can be wrapped in large vesicles. Baculoviruses infect in what are called occlusion bodies, where you have lots of virus particles in them. We talked about a paper here on TWiV a long time ago. Human T-cell leukemia viruses accumulate in what they call biofilms, viral biofilms, for cell-to-cell transfer. For polio, Julie Pfeiffer's group has shown that gut bacteria help put multiple particles together so they can infect cells at the same time. So there are lots of examples of more than one structures that can let more than one virus get into a cell. And their question is,
there was a paper that came out this summer that I linked to in the show notes about vesicle-cloaked virus clusters in norovirus.
Yes.
And I remember thinking it was really fascinating, and I couldn't understand how it would be advantageous. So this paper helped me out with that a bit.
So that's the question. Why would this be advantageous? And they say and this is a wonderful phrase, multivirion propagules should be disfavored because they reduce the ability of the virus to disperse. And you think about it, when you have single viruses, right, you'll be able to infect more cells. Whereas when you put hundreds of them together, you're going to have less of a chance to infect, unless they say there is some advantage for these multivirion propagules. There has to be something that would offset the cost of not being able to hit as many host cells. And the Allee effect, I presume it's pronounced Allee. Does anyone know? Is it Allee? It's not French. I
don't know. But it's spelled Allee.
It's defined as a positive relationship between fitness and population size or density. All right. And it has been shown for animals and plants and Vibrio.
Allee wrote a book called Invasion of Animal Species quite a while ago. He was, I think, at the University of Chicago, as I recall.
So you know the name.
I do.
Good. Well, they, in fact, it's been shown in entomopathogenic nematodes to have an Allee effect, but never shown in viruses. So they're going to address this in this paper, and they use VSV because you can aggregate VSV experimentally using saliva from some people. Now, Kathy, is this something that you used to use at all in your days?
No, I never heard of it. But
never heard of it.
You know, the first thing I thought of course, is that this is a virus of cattle, and so it's the vesicular stomatitis virus I think of as being in the saliva. So that didn't surprise me, I guess.
The first thing I thought of is how often I've wanted to spit in my experiments.
It's a common joke in my lab. Did you spit in that? Is that how you got that result? So
yeah, right. Well, here in this paper, and if Alan were here, he'd make a title based on the spit, but we're not clever enough. But now this gives justification to spitting in your experiments. So the simple system to take VSV and they insert a red fluorescent protein or a green fluorescent protein coding region into the genome. And now they have these two viruses that are marked and can be distinguished in co-infection assays. And of course, they can also be measured by plaque assay. And so you can imagine you can aggregate one by adding saliva and leave the other one monodispersed, as they call it, or not aggregated. Mix them together and see who wins.
Mm-hmm.
It's really beautiful. So if you aggregate the green virus and leave the red one monodispersed, 71% of the fluorescent areas in the infected monolayer. So now you can look under the microscope and see how many green and red areas there are. So if you aggregate the green, you get 71% of the area corresponding to green. And if you do it the other way, you get mostly red.
That actually blew my mind, that it would fall out that way. I wouldn't have predicted that.
No, I wouldn't.
Amazing.
And they have this. So the key is they have the same number of particles in both the aggregated and the non-aggregated, right?
And the pictures are absolutely striking.
Mm-hmm. They're very pretty. Yes, clearly. So basically, for an equal input, aggregating it increases fitness in these competition assays.
Mm-hmm. One thing that was not clear to me in these experiments is exactly what the multiplicity was. I tried to figure that out. It's low, I would assume, significantly less than one infectious unit or one particle per cell in order for this to happen. I also would have actually appreciated, they have something like this down the road, but not quite the fashion I would do it, an experiment where they varied the multiplicity. And I'd like to see this experiment done where you vary the multiplicity.
I think that's their idea, that the aggregating is increasing the multiplicity, right?
Yeah.
In the end.
But if you see, I don't know if you'd get this effect. Well, I don't know what the multiplicity was. I assume that it's significantly less than one.
Yeah.
But I don't know that.
Yeah.
Well, they say here we could do a calculation. They start with 10 to the 9th plaque-forming units per mil, dilute it 1 to 10 in saliva,
And then another 1,000-fold.
And then another 1,000-fold.
But I don't know what they're adding
from
it
10 to the 9th.
They use 200 microliters of that and add it to something.
Yeah, you don't know how many cells, right? That's what you need to know. Yeah.
I see what you're saying. Yep. We know the numerator, not the denominator. Okay.
So if you look in a growth curve type of experiment where you take time points and measure virus production, the aggregation shifts the growth curve to the left. In other words, the cells infected with aggregates make virus more quickly than single virus particles. They're left shifted. And I have to say that they make this lovely statement. We titrated supernatants by the plaque assay. So they got it right. They use titrated, and they're using plaque assays, even though they could use some other, they could do flow cytometry or something, but this is the best way to do it. Let's see what, yes. So this very clearly enhances production in a growth curve earlier than, if you use monodispersed viruses. You can't even detect viruses being produced at a time when the aggregated viruses. So they say that aggregation accelerates viral reproduction in the first infectious cycle, and that gives you an advantage over the non-aggregated viruses. So this is something that they were looking for initially, right? What does aggregation do? And you can see here it accelerates the growth curve.
And that acceleration seems to be kind of particularly relevant in the first 6 hours.
Right. The next question is, does aggregation reduce the number of infectious units and dispersal capacity? So they co-incubate the red and the green. So they take red, GFP, and green, and they co-incubate in saliva. So they're mixing the viruses and then aggregating with saliva, and they infect cells, and they find that a large fraction of the foci produced in this way are positive for both GFP and red, both green and red viruses. So in other words, as you would predict, the aggregates are giving to single cells both green and red viruses. So we're delivering multiple genomes. But you do not get a reduction. Aggregation doesn't give you a reduction in the number of foci.
It even gives you an increase.
It increases it. That's
from
right.
what I can see.
Yeah.
Which I would not have predicted.
No, I wouldn't have done that. No, that's really remarkable.
Yeah, that is really remarkable.
That's the Allee effect, right?
Yeah.
That's
right. Right. Before we forget, when I was having to look up what the Allee effect was, and they were talking about different examples of it in the introduction, I thought about the fact that when we plate tissue culture cells, we always say, well, you want to have them dense enough so that they're near their friends because they seem to do better. And it's all kind of apocryphal, but I mean, somebody presumably has done the experiment to show, how fast something doubles when it's densely plated and not. But that's where I went with it.
Yeah. Yeah. Yep. All right. Next, they look at the multiplicity of infection, and their idea is that aggregation of viruses essentially increases the multiplicity of infection. In other words, the number of virus particles that can get into a single cell. Makes sense, right? If you aggregate and the whole aggregate gets taken up into the cell, you're effectively increasing the MOI, right? So they inoculate their cells. By the way, they're using murine embryonic fibroblasts to do these experiments, and they use a range of particles to approximate a very low to a very high PFU per cell, up to 50, same as the Mali virus that I complained about. And then they determine viral titers so that you can see the per cell production during one infectious cycle. And basically the virus production, so without an Allee effect, the virus production should decrease as you increase the MOI because you're getting competition and you can't make, there's only so much cellular resources available. So beyond a certain point, putting more viruses in isn't going to help you. However, they found that the production of virus was always higher at an MOI greater than 1, and it always paid off to invade with multiple particles.
Mm-hmm.
There is a peak beyond which you don't get any more advantageous reproduction, but it definitely goes up with MOI.
Does anybody, Kathy, what's
Mm-hmm.
how long what's the time scale of a VSV
Oh,
growth curve?
I don't exactly know. Trying to remember how fast we could even see plaques. I think we would see plaques the next day.
Wow.
Because
So I think-
their early time point here, and they're looking for progeny is 4 hours.
Right. Right.
Mhm.
Well, yeah, they did say that when they were looking at the boundary of the first infection cycle between 4 and 6 hours post-infection. Right.
Okay, so the implication of that is that 4 and 6 hours is at least you can get through a whole replication cycle in that
Mm-hmm.
time.
Mhm.
Okay.
Yep.
So one thing that could be happening here is a phenomenon called genetic complementation or multiplicity reactivation. If you have viral mutants that on their own are defective, but together they can both provide everything that's needed to replicate, complement, or multiplicity reactivation was called because at a higher multiplicity you would see this phenomenon where you get multiple viruses infecting a single cell. So they asked, is this what's going on? So they mutagenized their virus population with a mutagen 5-fluorouracil, and then they did competition assays with these mutagenized populations. So you can imagine you mutagenize, you aggregate, and you see if there's any benefit over a non-aggregated mutagenized population. Didn't help to aggregate.
Right.
No help. So you're not just doing complementation. You're doing something that's independent of the mutational burden of the population.
In fact, the mutated aggregated cells seem to infect a bit worse.
Right. Okay, so what's going on? A few more experiments to sort of tease this out. So they looked at a few different cell lines. We've so far looked at MEFs. We're going to look at baby hamster kidney fibroblasts, BHK21.
And those are sort of the garden variety host cell for growing
For
VSV.
yeah. And these are these transformed, but what are they? Are they immortal?
Oh, good question. I don't recall.
I believe they are. I think they say it here
Because
that
they
they are.
say, yes, they're highly abnormal tumor cells as opposed to MEFs, which are not, right? They're highly
These guys
permissive.
go back 50 years or
They
so.
do,
They've
they
been
do.
around forever.
They do.
Yeah. And you're asking me questions about VSV, and I just did the calculation. You know, I worked on it for 2 years, 37 years ago.
All right.
So,
I'm
And
sorry.
you
I
don't
thought-
remember everything?
no, it's amazing, but I
sorry,
don't.
Sorry, we assume.
No, it's okay. It's okay.
I know that it's like it's a long time ago. I know it's
Yeah, and I'm happy to admit when I don't know it. So,
yeah, right. All right. So they do their same kind of assays, competition analysis of focus production. Saliva aggregated particles have a fitness advantage in BHK, but it's weaker than in MEFs. And the effect is compatible with an Allee effect. It's less so than in MEFs. And so they say, okay, there's a cell dependence of this Allee effect. So what is good? They look at a whole range of other cell lines, mouse and human and Grivet cells as well. And they do competition analysis with their two viruses. The aggregation is beneficial in some and it's detrimental in other kinds of cells, which is interesting. So the nature of the cell makes a difference in the Allee effect. And what they find is that aggregation is more beneficial in less permissive cells. Kind of makes sense, right? So for some
kind of makes sense.
virus doesn't grow well in a less permissive cell, but if you put more virus particles in, grows better, replicates better. Exactly what's going on, we don't know, but you can imagine delivering more viral gene products, is overcoming some deficiency in the cell, something like that. So that's kind of interesting. And now we get to Brianne's experiment here, where they ask, does it have anything to do with an antiviral response of the host cell? So they focus on innate immunity. They have a viral mutant, which has a amino acid, has a deletion of a single amino acid in the viral matrix protein. And normally the matrix protein inhibits expression of cell genes, and this mutation abrogates that inhibition. So this virus, Delta 51, is attenuated in normal cells. It doesn't grow well in normal cells, but if the cell has defects in innate immunity, it will grow pretty well. So they can look at the yield of wild type and mutant and ask, does aggregation have any relationship to the innate immune response of the cell? So again, they use their two-color viruses, aggregated or not. Aggregating increases the chances of overcoming innate immunity. So if you have even this Delta 51, you can overcome innate immunity with aggregation. That's a cute little experiment using that mutant, I think, is cool.
It is a really neat experiment. I ended up down a bit of a Google hole here again. And so for any, and they get to this a little bit in some of the next experiments that they talk about, although they are not in the, they're not shown in the figure when they're looking at transcript. But if anyone is wondering, the innate immune proteins that are bound by this matrix protein are not well described, and you can Google for a long time trying to figure out how exactly this innate immune block works.
But I got the impression that it was a general transcriptional block, right?
It looks like part of it is transcriptional, part of it is mRNA export at the nuclear pore, but there was also some other parts that were unclear.
But it's not specific for innate immunity. Lots of genes are involved, right?
I think so.
Yeah, that's my remembering of this. I'm not going to ask Kathy. She probably won't remember.
No, I've never heard of Delta 51 before.
Okay. The other experiment in this area, they add cytokine. They infect cells, and then they take supernatants, which would then have interferon-induced proteins in them, and they then add them to cells to sort of prime them with cytokines. And this is going to inhibit the replication of virus. But if you aggregate the viruses, you can overcome this pretreatment of cells, this inhibitory pretreatment of cytokines.
I thought this was really interesting. Before they did the pretreatment experiment, they also did some qPCR to look at when interferon-stimulated genes were made in the MEFs after infection. And they saw that the interferon-stimulated genes were actually made after the time points where this was important. And so it didn't look like the cell itself was making a particular ISG to block infection. This is happening too early for that.
That's right.
But they show that instead it's about when cytokines are produced to potentially affect the next set of cells. And so the idea that I got from this was that the virus was replicating so fast that it could sort of beat the cytokines to the next cell.
That's right. That's absolutely right. Because you're right. It's too early to happen. This Allee effect is happening in the first hours of infection. It's too early for a cytokine to have an effect on it. But it's in the next cell. Make a ton of virus, and then you can overcome inhibition in the next cell. Exactly.
Right. And I thought that was a really interesting model.
Let's see. Faster growth occurs in the absence of saliva. So I think maybe a reviewer asked them.
Oh, that's exactly what I wrote in the margin.
Me too.
This is an experiment for the reviewers.
Yes, whether the fitness advantage could be driven by some component of the saliva promoting infectivity, right? And they say, well, we don't think so because we dilute over 1,000-fold, but we'll do an experiment. And we can heat viruses at 37 degrees. If we have high titer stocks produced on gradients, we can heat them and they'll aggregate. And in fact, this increases viral fitness as well. So there's no saliva here at all, but the effect is less than with saliva because in part the heating destroys infectivity. And I thought it was fine. And then at the end, they say we can't really compare these two, so we can't formally discard the possibility that saliva might contain a proviral component that associates with viral aggregates, which is, I don't know. I don't know if they made them say that, because I thought the data were pretty good that it's not in saliva.
Just conservatively written.
So there you have it. There is an Allee effect for viruses. So aggregation provides clear benefits that would overcome the lack of ability or to infect more hosts with single virus particles. And the first thing I thought about was, we always look at populations of people, they get infected with viruses. Some people get infected, other people don't. And maybe this can explain why, because if one person gets many more particles, that could have an aggregate Allee effect and lead to infection. Whereas if you got a few particles, you're not going to get an Allee effect, and therefore you're never going to get infected. So this could have in vivo relevance, I think.
And I think you mentioned this in the beginning, but to come full circle on this, this would help explain on an evolutionary scale the findings for several viruses, in particular recently, that viruses are produced in bags of virus. Okay.
Right.
Hepatitis C and several others. Some of the large DNA viruses are produced in occlusion bodies that will have the effect of essentially aggregating the virus to deliver it to a host.
Yeah, this provides a reason why aggregating might be beneficial, right? Because those papers that you cite, they say, okay, viruses are aggregating, but we don't know why they would be beneficial. This explains it. And then they say at the end, well, let's do this experiment with other viruses now. Right.
It also, I don't think we ever mentioned the number, but when the peak per capita production was somewhere around three to four viruses per cell, depending on what time point you looked at. And it reminded me of the pseudorabies story, something about 7 viruses getting in per cell.
Yeah.
Yes, that's right.
These numbers are not very big. And then sometime within the last week or two, someone said something in a seminar about, the effective dose, or you can infect with as few as 7 virus particles or 7 plaque forming units. And I always think, why isn't 1 enough? And so maybe this is an explanation for that. At least the next time I hear that, I can think about the Allee effect.
Yeah. I think, though, this is a lovely experimental system because the aggregation is easy to do, right? Mm-hmm. I'm not sure for other viruses it's going to be as straightforward, but people will figure out ways to do it, right?
The other thing, toward the end in the discussion, they talk a little bit about the PFU to particle ratio, which, of course, I always think of as the other way around, but doesn't matter. Mm-hmm. But they say that there's potentially a lot of particles that are not infectious. And then they mention other physical methods to enumerate virus particles, such as electron microscopy, nanoparticle tracking analysis, or tunable resistive pulse sensing, which I had never heard of. I didn't have time to look it up, but there's other ways.
Yeah, there's a paper describing those, So that was an interesting discussion there, the particle to PFU. They say in previously published work, VSV infection of BHK21, it's been suggested that the PFU to particle ratio is close to 1. And the reference for that is a paper where they use nanoparticle tracking analysis, right? And what that means is that most particles can infect cells, and hence that no critical Allee effect could be operating because you only need 1. So how could there be an Allee effect? In contrast, here we have estimated that this ratio is on the order of 0.1 for VSV and BHK21 and as low as 0.01 in MEFs. So they're finding their particle to PFU is much lower than that other study, and hence that's why they see an Allee effect. Right?
Yeah, PFU to particle. For those of us who think of it the other way around, 10 particles per plaque forming unit or 100 particles per plaque forming unit.
Yeah.
So,
yeah. Makes you start to think a little bit about all of the emerging literature on defective interfering particles. and how this might be related.
So when I was in college and graduate school, we had a presentation by someone who was reviewing Allee's work, and one of them involved the use of goldfish. A single goldfish in a tank of water exposed to X amount of toxin died, and 10 goldfish in a larger volume of water to match the 10 times the volume of water of the first fish, but the same concentration of toxin per fish, all 10 lived, because they acted as a group and secreted mucus, which neutralized the penetration of the toxin. So he did a lot of crazy experiments that were, in the long run, all validated by examples in nature.
Oh, that's cool. That's an Allee effect at the level
first.
Of a fish.
And by the way, he ended up as the dean of the School of Sciences at the University of Chicago. Amazing guy. Amazing guy.
Dickson, that sounds like altruism.
It does, doesn't it? There's a safety in numbers. That's where the old expression comes from, the safety in numbers kind of thing.
Yeah, sure.
You know, don't go out on the streets alone at night. Make sure you're with somebody else that can fight off the invaders, whatever you're interested in fighting off. And that derives from a natural. You see lions attacking a single Cape buffalo, the buffalo loses. But as soon as the herd gets wind of the fact that one of their kind is in danger, they show up and scare off the lions, and that really is a good example. So we have, even in virology, you've got the best examples of the same effect.
I think recently we did. I can't remember which podcast, paper showing viral altruism. Oh, this is, yeah, that was on TWiV. This was,
it's kind of an anthropomorphizing.
For phages. So many phage genomes encode anti-CRISPR proteins. But very much like this Allee effect, when you think about it, when a single phage infects a cell, it can't make proteins quick enough to avoid destruction by CRISPR, because all the CRISPR proteins are on all the time. And the key is that you have to have multiple phages infecting a cell.
Very interesting.
Same idea, right? And they said in that paper, this is the first example of true viral altruism.
So this is an evolutionary characteristic that must have started way back when they were just cyanobacter. Yeah. So finding a Mimi virus in 30,000-year-old soil sample is not a big deal, I don't think. To be honest, because they've burrowed into these amber deposits with lots of insects and stuff like this, and found remnants of DNA that date back hundreds of millions of years, basically. So it's an amazing world out there.
Well, I think finding big viruses is pretty cool, simply because
I didn't say it was cold, actually. It was in the permafrost.
Is that TWiV, the TWiV 187 rounding up the bees?
Yes.
Okay.
Yes, that's it. All right. We do not have email this episode because people were cooking their turkeys, I guess.
We had some few emails.
We had a few at the beginning. Let's do some picks. Brianne, what do you have for us?
Okay. The New York Times had the 40th anniversary of the Science Times this week. And as a part of their 40th anniversary celebration, they published 11 different articles on big questions in science that they thought needed to be answered. One of those is the question, why don't we have vaccines against everything? And it's a really nice article that talks about a lot of the challenges, both scientific and in terms of funding, in generating a lot of different vaccines. So I really liked that one. I thought it described some of the issues in vaccine development pretty nicely for a lay audience.
Cool.
In fact, it seems like with their 11 questions, they were very into the immune system.
Of course, you wouldn't want a vaccine against everything, right? I know that's not what he means, but you wouldn't want a vaccine against Mollyvirus Sibiricum, right?
No.
Don't need it because it doesn't infect us, but I got it. It's by our friend Don O'Neill, Dickson. Do you know Don?
McNeil. I met him.
McNeil.
Yep.
That's right. You met him.
Your friend Donald, what's his name?
Well, he's not my dear friend. No, not at all.
No, but I think you should have him on sometime. I think you would find him really entertaining and pretty good.
Yeah, we could. He's very hard to pin down. I tell you, we tried to get him. Oh, so he published a Zika book a couple of years ago, right?
Yes.
And his publicist emailed me and said, could you have him on to help push the book? I said, yeah, we could not get a time that worked for him. Because he said, I'm sorry, I travel a lot, and but we'll try again.
Yeah. Well.
Rich, what do you have for us?
So occasionally I have picks related to sailing that I usually justify as technology. If anything, this is anti-technology. A race that I picked some time ago is called the Vendée Globe, which is where it's a single-handed, round-the-world, non-stop, unassisted sailboat race. They start off in France and sail south and then east around the Cape of Good Hope and south of Australia and Tasmania and New Zealand.
Wow.
And Cape Horn, and then back up through the Atlantic to France. That's the Vendée Globe, and that's based on the race I'm picking here, which is the Golden Globe race. The Golden Globe race was the first such round-the-world, nonstop, unassisted, single-handed race that happened in 1968 and 1969. And the winner was a gentleman named Robert Knox Johnson, who was the only one who finished the race out of the 9 guys who started. But since that time, they've run it about every 4 years, or at least that's what they do recently in the Vendée Globe. The technology, of course, has evolved enormously. So they've got satellite weather tracking and satellite navigation, and these boats that are like 60-foot giant surfboards where they can make water. They have desalinating devices and electricity generators and all of this crazy stuff. And the Golden Globe race is basically saying this has become much too technical. And it's about the technology now and not about the sailors. So let's go back to the beginning. So this is running the same race, and the rules are that you can't use GPS. Okay. You have to use 60s technology, including a 60s or actually earlier than 1988 boat style. Okay. So these guys are going in boats that are between 32 and 36 feet that were produced in the 70s and 80s. They're using all just what we call dead reckoning navigation. They don't have GPS or anything like that. They're using charts and compasses and log lines. They can't make their own water. They have to trap rainwater. And it's amazing. So I've given the website here for the race where you can track it. We're now 145 days into the race. Which is what, 4.5 months, almost 5 months. They've got another 2 or 3 months left. The leader is like 6,000 miles ahead of the trailer. They started with 17 boats, and they're down to 8. Okay, so 9 people have dropped out for one reason or another, not the least of which is 3 of them actually rolled over in the Southern Ocean and were dismasted and had to be rescued. Matter of fact, the leader's in trouble now because he was in a storm a little while ago and pitch-poled. That's going forward end over end down a wave. Okay? And so he has damage to the rigging on his mast, but he's decided that he's going to try and, rather than quit, he's going to try and finish the race with that. He's rounding Cape Horn almost as we speak now. It's just an amazing event. Yes.
Explain to me, they list the top 5, and number 4 is Susie Goodall, who's a 29-year-old woman from the UK who's been sailing for 26 years.
Yeah.
But she's in 4th place, having sailed 17,000 miles, and the 3rd place person has only sailed 16,000 miles.
Are you looking to distance to finish?
No, total nautical miles sailed under the leaderboard here.
Okay, so sorry here. We'll have to go to this. What I really like is the live tracker. There's a sub-site in this where you can see a map of where they are, and you can play with it, and you can superimpose the weather. If I go to the leaderboard, and you mentioned Susie, which is thank you, because she's my favorite. She's the only woman in the race, and I want to see her. I mean, she's way behind now, but anything could. I mean, she's not way behind. She's doing very well. She's in the middle of the pack, but anything can happen. This is survival. Okay, let's see here. Distance sailed 17,000 nautical miles. The leader has sailed 19,000.
But she's behind number 3, who's only sailed 16,000. So maybe that's just some typo.
Wait a minute. Number 3 has sailed 16,000. Oh, okay. It could be that I don't know. Yeah, I don't know what's going on there. It could be that those distances include some sort of excursions north and south of the run line. Could be that sometimes it takes longer to get where you're going, because they have this other thing is velocity made good, which is really a complicated formulation. I don't even understand it, but it's supposed to be a better representation of the progress that you're making. At any rate, have a look at it. And I recommend this thing if you get into it, the live tracker. They can tweet. They get minimal tweets, 100 characters that they can send off on a daily basis. And they have a weekly safety satellite call. Okay, so once a week they can talk to their team and make sure everything's okay.
And those are posted on SoundCloud, but you can get to them from each of their sites. I mean, each of their sub-sites from this page,
but I like looking at the live tracker and superimposing Windy, which was I discovered in the Golden Globe race, which is a weather app that I actually wound up picking on another TWiV, and see what sort of trouble they're about to get into. Looks like this leader he's actually around the horn, I think, and that will get him out of the crummy Southern Ocean weather. So,
Rich, if they get in trouble, what happens? Is there anybody nearby? Is there anyone nearby?
They do what they can. I mean, these guys that recently got in trouble were 2,000 to 3,000 miles away from any help. And so what happens is that they are not too far from some shipping lanes. Okay. So an alert goes out. The alert goes out to the other sailors. So if there's anybody nearby, they may go and see if they can assist. In these other cases, they wound up being close enough so that they could barely send a plane out and drop some divers and rafts. But I think that was only after there were some rescue ships close enough by. It's tough. It can take them days to get to them. And sometimes it's with commercial vessels and sometimes with rescue vessels. Do
people die in this race?
Well, not this race so far. People have died in similar races, yes. Not uncommon.
What's the hazard from icebergs?
They have several places you're not supposed to go south of for that very reason. And I think that certainly in the Golden Globe, in the Vendée Globe race, they can see the icebergs and on electronic devices.
Oh, sure.
In this particular race, I assume, but I don't know, that if there were a serious risk of icebergs for any one of these boats, they would send them an alert. Okay, but I don't know that for sure. It is an issue. It can be an issue. This guy who just rounded the horn, right before he was rounding the horn, he was in what for this race in the Southern Ocean is moderate conditions, which was 45 knot winds, 45 knot winds and 6 meter seas. Can you imagine a wave after wave of 20 feet coming at you?
Yep. My wife Marlene went to Antarctica last year, and they had to go between Ushuaia and George's Island, and it was a very interesting tour.
The Southern Ocean is dangerous place to be.
It's the worst ocean on Earth. That's true.
Rich, have you ever pitched forward?
No. Closest I've ever gotten is what's called a broach, which is where you sort of you're wildly tipped over enough so that this one time, God, I was standing up on the deck flying the spinnaker, and all of a sudden I was halfway up. I'm standing on the deck, okay, and all of a sudden I'm halfway up to my knees in water, and I look back and there's water just flowing into the cockpit. And the skipper is white as a sheet, and the boat's going crazy, but it, righted itself and bailed out, and we made the adjustments, and that was okay. But these guys, that's their life. They're doing this all the time.
Can't imagine. I can't imagine.
It's crazy.
But I have zero sailing skills, so you have to have people who know how to sail. Would you do this, Rich, if they asked you to come with
No
me?
way. No way. No way.
Dixon, what do you have for us?
Yeah, well, I have a commemorative. I would like to honor a friend of mine. His name is Robert J. Demarest. He passed away earlier this week. He was 90 years old and was recognized as the greatest living medical illustrator of his time during most of his career while he was at Columbia, and we met and became very dear friends and good friends and fishing buddies and all kinds of other things. He helped illustrate a few of my books. One of them was the West Nile virus book. And his last book, which was published by himself about 3 months ago, called The Anatomy of Figure Drawing, Secrets of a Master Medical Artist by Robert J. Demarest. I highly recommend it for anybody that's interested in painting or drawing or whatever else. And then my second pick is the biography of Allee. And you get to see where he's been and what he did and why he was so revered as a scientist. Because he was in the 1930s, this was before computers, all kinds of other things, and he was making these remarkable mathematical calculations of populations and how they interact with each other. So those are my two picks.
Interesting. Allee was chairman of the zoology department at University of Florida at the end of his career.
Oh, I didn't know that.
And met his demise in Gainesville, Florida in 1955.
Isn't that interesting?
He must have. Yeah,
because Howard Odom ended up there too, and he must have been a good friend of his.
Dickson, he illustrated your book on flowers and poems, right?
Yes, he did.
I remember he came here and you were working with him. And he made little.
Yeah, no, he was a wonderful guy, and very sad. He had a mitral valve defect that didn't actually manifest itself until he turned 85. And the way he found out was he and his wife went to Mexico City last summer. And the moment he got off the plane, he couldn't breathe. So he said, something's wrong. We have to turn around and go home. And they did that right at the airport. And 3 hours later, they went on a flight out back to New York. And he immediately went over to Columbia, and they diagnosed it. And they said, you only got 80% of the amount of oxygenated blood being pumped into your body every time your heart beats. You've got a problem here, and they tried to fix it, and it was, of course, a very risky operation. The operation was successful. The microbes got him, unfortunately.
Yep.
All right, Dickson, he's got an acknowledgement to you and his dedication on this book.
Yeah, he's a wonderful guy. Wonderful guy. It's just I just miss him so much already, and he's only been gone a couple of days, so you know how these things go.
I have one of his books here in my office. I can see it. I can't read it. It's something about Homer, Winslow Homer.
Yeah, traveling with Winslow Homer. That's right. He got involved very early on in our friendship with trout fishing, as you might imagine. So we began to go trout fishing a lot. And I said, "Are you familiar with the paintings of Winslow Homer?" And he says, "Yeah, kind of." And I got a book of his early work that he did up in Maine. And he fell in love with it, and he just took off on it and ended up going to all the famous places that Winslow Homer lived at and illustrated a book basically dedicated to Winslow Homer's travels.
It's cool. Yeah, I remember that. Same. Well,
yeah, and the thing that was really controversial about his work was that he published a book on the fetus as it develops in utero. Mm-hmm. And each picture was an absolute size of the fetus at the time per page. And then at the end of the book, he had a whole section on contraceptives, and that, he said, that killed the sales of my book.
Yeah.
And he was fierce and determined not to give in to that.
Yeah.
And he did. He's published it the way he thought it should be, and so I have deepest respect for him.
Kathy, what do you have for us?
I have something. Sometimes I get these things from Jason Kotke, and the gist of it is that depending on which hemisphere you're in, the moon looks different. And I've been in the southern hemisphere, and last night I think was pretty much full moon here. So I was looking at it through a skylight and then trying to remember what I saw when I was in Brazil. But the gist of it is, the title is "The Moon is Flipped Upside Down in the Southern Hemisphere." But of course, it's not really flipped upside down. It's just a different orientation, and it's a simple picture that shows you why it is. And what Jason Kotke says is, "How old were you when you learned that the moon in the Southern Hemisphere is upside down?" I was today years old.
Yeah, right. That's me.
That was me too.
That's fine.
So I just thought it was some surprising thing that I never knew. And there you go.
Did you see it come up last night, Kathy?
I didn't see it come up in the middle of the night. I was awake.
It was astounding. Now it came up here around 6:30 or 7 o'clock, and it was orange. It wasn't white. It was, and it was absolutely stunning.
Yeah.
Well, now I'm going to have to remember next time I'm in the southern hemisphere to take a picture of the moon. So I'll have it for comparison.
Well, it's hard because the moon is really bright. So I looked at my picture of the moon from Brazil, and it's just this bright white dot. So good luck with that.
You're going next year, right?
I'm going at the end of December.
Yeah, there you go.
The other remarkable thing, if you can see it, Brianne, is the end of our galaxy.
Okay.
So I was actually in Alice Springs one year, and we took a trip out to Ayers Rock, and halfway back, it was like 2 o'clock in the morning, and the bus driver emptied the bus, woke us all up and said, "You got to come out and see this." There wasn't any light pollution whatsoever, and you could see the actual end of our galaxy. And in the
That's
two
cool.
Magellanic Clouds, it was the most remarkable sky I've ever seen.
All right, my pick is a new illustrated children's book about viruses and vaccines, and it is available for free in both English and Spanish. In English, it's called Paul Has Measles, and in Spanish, Pablo Tiene Sarampion. And it is put out by a friend and colleague of mine, Susana Lopez, with her.
I'm going to buy this.
It's free.
It's
I'm going to download it for free.
Download it and share it. Martha Yocupicio, Selene Zarate, they're all virologists working in Mexico, and they worked with a graphic illustrator, Eva Lobaton. And their goal was to give this away, and they wanted to print it, but they ran out of money. But eventually they'll print it and give that away. But in the meantime, I'm helping them to distribute the PDF.
Oh, that's great.
And it's something that, kids should read and understand what viruses are and why I need to be vaccinated and so forth. So I put the post and the links on the blog, Virology blog, so check it out. You should download it.
The illustrations are fabulous, and I sent this to about 25 former lab people this morning and to my current lab, and the adenovirus has 3 eyes. I love it.
Yes, the viruses are great. Yeah. So they're also recording a version, a little movie, right? So they'll put it on YouTube because you have to be there as well to get it out. And I told them I would do one in English. But maybe, Kathy, you'd be interested in doing the little girl's voice.
Sure. Yeah.
Because I need, because it's Pablo and there's another.
There's a girl. Yeah, I don't
there's
know.
A girl. Is it just
her
one?
name.
But we can connect on how to do that, and then I'll just put it to the pages turning. I should probably get a third male voice to do.
Oh, it's Sophie.
Sophie, right? So we have Paul's mother. And then we have, Sophie. So, yeah, I'll be in touch.
This is brilliant. I really love the pictures.
Pictures are
Aren't
great.
they great?
Fantastic.
The curly purple hair of their teacher.
I like the illustrations of the microbes.
Microbes are great, yeah.
Yeah, is there a pox virus? I have to go back to that page.
No, I didn't see the pox virus. I didn't see
yeah, maybe there's not. But there's definitely a rhabdo, and the adeno with 3 eyes.
So I helped to edit the English version. They had sent me because they had some funny,
syntax. Yeah.
Yeah, and also the names. They were. What kind of name should we use that would be popular or understood in all languages, right? So because they were going to, they would use a Spanish sounding name, but it might not necessarily be good here. So I like it. It's very good. It's a great idea to get kids to learn a little bit about viruses and vaccines, and I love that they're going to give it away. So hopefully that will help. So share it. Everyone who hears this, share it on all your social media and so forth. Send it to your friends. The more people, the better.
Page 21, the picture of the, if our cells are sick or get broken, we get sick because the viruses get out. And it's just such a great illustration.
Yep. Yeah. It's well done. All right. That is TWiV 521. You can find it at Apple Podcasts, microbe.tv/TWiV. It's also on a lot of other places like Stitcher. It's on, what's the big music sharing service?
Pandora.
It just came out on Pandora. So TWiV, in fact, was...
How appropriate.
Pandora's, yes, Pandora viruses. Pandora selected us to be part of their rollout, which is cool, which is, I think, happening in December. What's the other one? There's another music service.
Spotify?
Spotify. So you can get it on Spotify as well. And also Google Play. Someone emailed and they said, you're not on Google Play. I said, yeah, there's a link in the show notes. If you have an Android and you want to listen on Google Play, you can do that. But of course, if you have any podcast player, you can listen. Just subscribe. How do you subscribe? Well, you could search for This Week in Virology and you'll find it and get every episode. And subscriptions help us so we know how many people are listening. That really helps us a lot. So please subscribe. And of course, if you like what we do, consider supporting us financially, microbe.tv/contribute. Dickson Despommier normally is here in front of me.
Normally.
But he's not. He's abnormally away.
I'm abnormally in Fort Lee.
And he is, he can be found at parasiteswithoutborders.com and Racaniello.org. Thank you, Dickson.
So always a pleasure, as I will make up for Alan's absence.
I appreciate your tenacity in trying to get online.
Oh, just downloading this stupid new Skype was absolutely painful, but it worked.
Okay, thank you very much. Brianne Barker is over at Drew University. She's on Twitter as BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here.
Thanks for dialing in from upstate New York.
Yes, it's a little colder than my usual podcast location, but we made it work.
When are you coming back? Sunday?
I think Saturday night.
All right. Safe driving.
Thank you.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Got a big game coming up tomorrow, Kathy.
Yeah, not here, though.
No.
That's good.
In the cornfields.
Which game is that, Dickson?
Ohio State, Michigan.
Oh, yeah?
That's a big one.
It's a big one, yeah.
Oh, yeah.
It's for the Big Ten championship.
And a place in the playoffs. That's right.
Yeah, that's what I mean. Who gets to play in the Big Ten championship, I think is how it goes. Yeah.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Thanks all of you for doing this the day after Thanksgiving. Year after year, you do it, and nobody complains. Thank you very much. It's great. I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASM for their support and Ronald Jenkins for his music. And please go to youtube.com and subscribe to my videos. Search for Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.